You probably already know that RNA sequences are translated by ribosomes to make proteins, but some RNA sequences form structures of their own by folding into secondary and tertiary structures. This folding is driven primarily by the fact that certain nucleotides like to pair with certain other nucleotides. A G likes to bind with a C, and an A likes to bind with a U or a T. Each nucleotide can only bind with one partner at a time, and long stretches where each nucleotide has a binding partner elsewhere on the sequence are favored over short stretches or individual non-sequential pairing. These pairings compete with each other since any G in the sequence could pair with any C, and similarly any A with any U or T. While a given final arrangement may be optimal in terms of maximizing the length of coherent runs in paired nucleotides, these arrangements are only probabilistic, and outside forces such as temperature and pH may induce a different pairing arrangement. The molecule may even transition randomly between many different nearly optimal structures based on small fluctuations in available energy. Here we see an RNA dot plot of our sequence and one of the possible structures that it could fold into. These dot plots are calculated by RNA folding algorithms and are then used to generate a two-dimensional schematic, seen at the left. This is a flattened picture showing how the nucleotides arrange themselves in pairs. Looking at the upper right triangle of the dot plot, we see dots of 4 colors, where each color scheme represents one potential folding configuration. The structure shown here was plotted using only the red dot color scheme. In nature, different parts of different binding schemes may in fact be interchanged. Let's walk through the dot plot and see how the plot and the structure are related. In this example, we're going to focus on the red dot color scheme used to generate the folding configuration seen at the left. We start at the 5' end of the sequence. Position 1 of this sequence is a G and is shown at the top of the diagonal on the plot. From that position, we read across to the right. There is a green dot to the right, meaning that in one possible folding scheme, base 1 does have a binding partner somewhere else in the sequence. The structure on the left, however, is derived from the red color scheme, and since there's no red dot, that means that in this folding scheme, the residue at position 1 has no binding partner in this structure. Now let's look at position 2. Again, there is a green dot indicating that in a different folding scheme this position has a binding partner, but no red dot, so no binding partner for the structure on the left. Now looking at position 3, when we move to the right of position 3, we do see a red dot. Moving down from that, we see that it is on position 62 of the diagonal. This corresponds to the third base of the sequence, binding to the 62nd base, which is in fact what we see in the structure at the left. In this fashion, we can read through the entire dot plot and determine whether or not a residue binds to another residue in the sequence. Again, recall that in this example, we're only focusing on the red dots. The other colored dots represent other structures that could be formed by this sequence. Let's go ahead and speed things up. I think you probably get the idea by now. It should be easy for you to see how a line of dots in the plot corresponds to a stem in the structure, and an area of sequence without dots corresponds to a loop. Remember that the similarly colored dots only represent what the software predicts to be the best foldings in the absence of any other information about the RNA and its environment. Environmental factors, such as binding with another molecule, could cause these structures to be non-optimal and make other structures more optimal. The rules of G binding to C, or A binding to U or T, as well as the sequentially collinear regions where good pairings occur in long stretches, are invariant of the environment. We can consider the other dots in the plot to be potentially important alternative pairings, even in the context of a supposedly optimal structure like the one indicated by the red dots. At position 16, we've run into a stretch of the dot plot where there are no red dots. meaning that these particular bases do not have binding partners in this scheme. Notice how this corresponds to a loop in the structure. Now we have a brief stretch where the bases do have binding partners. Looking at the dot plot, we can see a big triangle with a small triangle nested inside of it. This recapitulates the folding structure we've explored so far. A large paired stem with a region of sequence that's not paired, and then another small stem. The highlighted residues have already been consumed by their pairing with other nucleotides, so we skip over them to the next unoccupied residue on the diagonal at position 30. Now we traverse another stem and loop, and again the dot plot shows us nested triangles, with the distance between the smaller nested triangles telling us about the small unpaired loop at the end of the smaller stem. Let's finish up our mapping and then look at some of those alternative pairings. For example, let's imagine that something external induces positions 13 and 14 to not pair with positions 52 and 51. The dot plot tells us they could also pair with, for example, positions 21 and 20. Let's erase the pairings to 52 and 51 on the dot plot and see what happens. Because RNA-based pairings prefer to occur in multiples with other sequential neighbors, the lone 15 to 50 pairing is unlikely to remain, so we'll remove that as well. With 13 and 14 now paired with 21 and 20, the dot plot tells us we've created a problem because we'd previously used 20 and 21 in a different loop. So if we're going to use 13 and 14 to 20 and 21, we need to remove the pairings of 20 to 29 and 21 to 28. As before, the lone 22 to 27 pairing becomes unlikely, so it gets removed as well. We're not going to go through all of the alternatives, but now we've left the entire green shaded triangle in the dot plot to be filled in with new possible pairings, generating alternative possible 2D structures. The function of a given RNA molecule may be determined solely by its sequence, or by the structure that it's most likely to fold into. It may also be governed by whether some small segment can fold into a particular substructure, even if that substructure doesn't appear in any optimal fold, or even simply by the diversity, or lack thereof, of structures that the RNA can fold into. Now that we've looked at how a dot plot can be read into a structure, let's look at some of the other possible structures that can be formed by this sequence.